Good afternoon, everyone, and welcome. I'm Cheryl Reynolds with the UC Statewide IPM program, and welcome to the UC Ag Expert Talk on snails and slugs. Peter Cosina is also here with me, and he'll be running our polls and doing any troubleshooting. And now I'd like to introduce our group of speakers today. So first we have Dr. Cheryl Wilen. She's an area advisor, an IPM advisor in San Diego. We have Dr. Rory Mc Donnell, an assistant professor at Oregon State University, Dr. Adler Dillman, an assistant professor at UC Riverside, and Dr. Irma De Ley, an associate research specialist at UC Riverside. And today they're all speaking on snail and slug biology, damage, and current research. And so first, I'd like to pass this over to Cheryl Wilen, who is our first speaker. So Cheryl, I'm going to pass this over to you, and you can go ahead and share your slides.
Okay, thank you. I'm going to get started on this. So just wanted to start out with some basics about snail and slug biology. The next presentation is going to be by Rory Mc Donnell, as Cheryl mentioned, and he's going to talk more about slugs, and I'm going to concentrate a little bit more on the snails. So, but to make sure we're all good about integrated pest management, first of all, we want to know why we want to do this. So, number one, we want to make sure that people understand that the idea behind integrated pest management is more of a long-term solution and focuses on prevention of the problem. And in order to do that, we have to know something about the pest and its biology and where it lives. So, and then the other thing is that we don't want to spray or apply a pesticide just because we see a pest. So an individual snail or slug may not be a problem, but if you see a large number of them, and I actually was just at a nursery yesterday where I saw thousands of snails coming in from a wasteland area, then we really have to consider other methods. And the other thing that we also want to consider is as part of integrated pest management is that we want to use the least toxic method to protect people in the environment. So, and by least toxic, I don't mean least toxic to the pest. I mean least toxic to humans, pets, wildlife, and water quality and air quality and so forth. Okay, so let's start right in with the brown garden snail. It's the main snail that we have a problem with in California, particularly Southern California. Imported snail, comes from Europe originally. You might know it as Helix aspersa, but the new name that's been accepted probably for at least 10 years is Cornu aspersum. We're trying to make all of our documents reflect that in the future, but you will probably still see Helix aspersa in some documents. So anyway, that's the one we're looking at, and the way it feeds is it uses a rasping tongue, which I'll show you in a second, and generally scrapes the surface of leaves, and then or often goes into the leaves or the rind of an orange, for example, and causes damage that's irreparable and lowers the quality of the produce or the commodity that you're working with. Very difficult to find sometimes. So, and they, we're at night, you feed at night often, so sometimes you might not even see some snails and slugs. One of the things that we're aware of is that the snails do need calcium in their diet, and they often get that from the soil or often in what they're eating, and that is so that they get a nice hard shell. Sometimes you might pick up a snail and it just crushes in your hand, and in that case that's an area where the snails don't have enough calcium in their diet to really be protective. It's good for us though, because it's very easy to just crush those types of snails very, very well. So snails versus slugs. Snails have a visible shell, whereas slugs do not have a visible one. They're both mollusks though. This part, I'm not sure if you can actually see my my arrow, but just to let you know that the two tentacles that are pointing up, or the antenna that are pointing up, are actually the eyes there. And so that's pretty interesting to see that. And then the other thing that we want to show you is that the mucus gland is internal, and that's what makes the snail trails, as it were. The tongue has radula on it, or excuse me, they have like rafts on it, kind of like a rasp that you would use on wood. And that's how the snail feeds. Essentially, it scrapes the surface of the plant or whatever it's eating. So, and so what happens is, like for this plant itself on the right-hand side, sometimes you will get some early damage that might not be problematic, but as the leaf grows, these cells break apart, and then you get this sort of this shredding of leaves as the plant grows. Snails, terrestrial snails and slugs are hermaphroditic, means they have both male and female parts to them. And so it doesn't matter who they encounter. Okay, they can mate with any other snail of the same species because they're not male, strictly male or strictly female. What we'll find in the ground once they mate and lay eggs is that we'll find what they call clutches of the eggs. And the picture on the left-hand side shows a freshly laid clutch of brown snail eggs. So they're bright white when they're first laid, and then as the eggs mature, they become darker and brown, like in the upper right-hand picture. In the lower right-hand picture, those are slug eggs. They're much smaller, and in my experience, and maybe Rory can tell me whether I'm right or not, is they're not really laid in groups like they are, at least the slugs that I see, like they are with the brown snails. And they can lay eggs, typically about 5 times a year, and they get anywhere from 20 to 80 snails. Eggs in a clutch. I haven't really seen that many. Generally, they're on the lower end, and they're pretty fragile. So if you can sort of pick them up, sort of cultivate the ground. If you do see these snail eggs as you're doing some walking around, once again on the soil surface, and they'll dry up pretty easily, and the snails inside will die. Okay, they do estivate, which means they will go up on a tree or sometimes a fence post or something like that. They create this paper shell over the opening called the epiphragm, and they'll just sit there until the conditions are good for them to move around and start feeding again, or go to the ground and start feeding again. And so we can get numerous snails on trees. Here's some on a citrus tree on the right-hand side, just hundreds and hundreds on trees sometimes. Sometimes they're very difficult to knock off because they're really stuck onto the tree trunks. And as far as management, we know that they're generally more active at night or in the cool mornings, anytime it's moist and cool. And we also find that they tend to travel back and forth. They have a place that they like to hide when it's warmer or hotter or drier. So they like to hide then, and then at night or the evenings they will move out to the places where they're feeding, which is kind of interesting about their activity. So ways to control them. Some growers have tried what's called a Bordeaux mix, which is essentially copper sulfate and hydrated lime, sometimes mixed with like some kind of sticker type paint, which paint around the trees. Places that I've seen, it's been very, very successful. It's a repellent, but nevertheless, I've found that once it's on a tree that we have a very good control of snails in the tree for an extended period of time. The other things I'm sure a lot of you have seen are the use of copper barriers. If you've got a large number of trees or other kinds of things that you're trying to protect, that gets kind of problematic. It's hundreds of these bands that you would have to put on, and it's also very helpful if you frill it somewhat around the trunk to also act as a deterrent for the snails moving up the tree. And then we have actually found that you can trap them out. You can really reduce the population by using traps, whether using a wood board or something else that's attractive to them. If you do that on a regular basis, you can really reduce the population. But by a regular basis, I mean, you really have to do it on a daily basis. All right, and then the chemical controls are baits or a spray. And so the baits right now that work the best are iron phosphate, which often you can get ones with an organic label. There's another one that's sodium ferric EDTA, which is also an iron-based bait, but it's not organic, but it works a little bit faster. And then the old standard current standard actually is metaldehyde. And that one, it works very, very quickly. And I mean quickly, as soon as the snail eats it, it'll start frothing and then it'll die very quickly. And it's actually pretty satisfying to see that, okay, because you'll see a bunch of snail shells. But there is concern that if pets do eat it, they can be poisoned. The iron phosphate's a lot safer, and the same with sodium ferric EDTA, but often you don't see the snail shells, so you don't really know how well it's working unless you see reduced injury, because the snails tend to stop feeding and then they become photophobic, so they don't like light, and then they'll go to a darker place and that's where they'll die. The other one is mesurol, and mesurol is methiocarb, so it's a carbamate, which can cause some nerve injury to applicators if not used appropriately. It's also a restricted use pesticide, so most labels say that you can only use it, I believe, twice in a growing season. A lot of limitations. We use it a lot in ornamental plants as a rescue treatment. So for example, if you're getting ready to ship and you find that you have snails on those plants or the pots, that's when you would use it. You can use it on non-bearing fruit and nut trees and vines growing in nurseries and greenhouses and so forth. But as far as food production, again, it's just non-bearing. And again, it is restricted use. So it's got a lot of other considerations about reporting to the Ag Commissioner and so forth when you use it. Natural enemies for snails, the main one that we use is the decollete snail. Keep in mind, it really can only be used in Southern California. It's not allowed to be used in, I believe, past Ventura. May be able to go a little farther north, but there are definitely regulations about where you can use it. So for those of you in other parts of the state, please don't think about taking it to your area of the state. We found that it actually is because it's a non-selective carnivorous type snail. We find that it feeds on native species, including ones that are rare or even often very in a critical state. We have an insect that also feeds on the snails. I have not found it myself, but I'm just letting you know that there is something that will do that. And the last thing I want to do before I move on to Rory is to talk about this snail called the Italian white snail. We're finding it is a very critically bad snail. Right now, it's in Los Angeles, but hasn't been found really in ag producing areas, but certainly in San Diego, it's really moved from sort of these areas that are just sort of wasted weed areas, sort of non-production areas. Now they're moving into production areas. I've seen it on avocados, very strong. Luckily, it wasn't a shipping avocado orchard, avocado grove. In nurseries, very, very bad. Right now, it's a B-rated pest, so the Ag Commissioner only will act on it if it's a shipping issue. And I think right now, as far as what I've seen yesterday, I think it's reached that point. So, and I do know of some crops that have been rejected after it's been found. In the interest of time, I don't want to go into this too much, but just keep in mind that we do know something about its life history and when the control should take place. But here's a picture on the right-hand side, is just to kind of show you the impact it can have. Really likes to just be on weeds. So mustards, for example, is what we have in the fall, and they grow up and they will feed on that. And when it gets hot, they'll go up on that. So whatever you can do to reduce those kinds of weeds in and around your growing areas will really reduce the impact of these snails. We also know that baits are not effective when they're small because they're eating vegetation. We really have to be very vigilant in when they're applied, and also keep applying as long as you can. Again, right now, the only place it's actually officially reported in the United States is San Diego County. 2017, all these red dots here on the left-hand side, that's where it was reported. 2019, so this year, many more reports and more impact. And there's been reports of it in Long Beach, more in the Palos Verdes area. And then looks like there's a little bit away from there. So I'm not really sure why it hasn't been officially reported in that county, but nevertheless, people should be aware that it is moving and can be found. So again, I think it's going to be a really, really bad problem for production because it's a very small snail. Sometimes it's not detected, and if you're packing, for example, citrus or avocado, and you find that they're very likely to get it actually more in the shipping container, okay, the crate, rather than the plants. So, oh, I do want to go back. There was a poll question I actually wanted to. One of the things I wanted to show you, hopefully you can see the screen. So you notice on the left-hand side, it's iron phosphate. You see that the bait is sprinkled broadly, whereas metaldehyde here, it's very, very heavy. We actually, even with metaldehyde, we want it to be a little bit more spread out than that. These products tend to cake when they're closer together, and they're not more attractive to the snail or slug when they're closer together. So save your money, do a little bit broader spray, follow the directions on there. Excuse me, broader application, follow the directions on the label. So don't make like a line of this stuff, even if you have a higher snail population, because again, when they get wet, they kind of mesh together, and then they're just not attractive enough for the snail and slugs to eat that. So
yeah, so Peter's going to bring that poll up again.
Okay, thank you. So considering the cost and damage, how would you rank snails and slugs as a pest?
And while they're answering that, Cheryl, I have a question from Richard for you. He asks, are there any endangered or threatened snail or slug species that might be affected negatively by current chemical control methods?
Yeah, so these chemical control methods are non-selective they're selective for snails and slugs. Measurol is not. Measurol is also used for thrips in the ornamental industry. But yes, so definitely any snail or slug that eats those baits will be negatively impacted and likely killed. So in fact, when people are using the decollet snails for their snail management, you don't want to use that in the same time that you're using baits, because they will also be killed.
Okay, so Peter's going to bring up the results to this poll.
Okay, so so many people consider that snails and slugs are in the top 5, and about that's half the people, and of those, and then about a quarter of the people think it's in the top 10 of their problems. So that's interesting to know. And it's a lot of people feel like 8% of the people feel like it's the worst. Another 8 feel it's like the second worst pest. So yeah, so at least we know that for many people it is snails and slugs are impacting their production systems.
Okay, he's going to bring up the next poll now.
Okay, so how should baits be distributed? And the selections are as a solid band, so snails and slugs will find the bait easier. Evenly distributed on the soil or media surface, about 1 to 2 inches apart. Or evenly distributed on the soil or media surface as close together as possible.
Okay, he's going to close this out in just a second.
Okay, so almost everybody got it right. There was one person that wasn't listening, sorry. So yeah, so it should be evenly distributed on the media surface, about 1 to 2 inches apart.
Okay, and then I think there's just one more poll.
Okay, which of these baits do you use most often? Do you use, and you can choose, looks like more than one. So the first question would be, so is it metaldehyde-based iron phosphate, sodium ferric EDTA, methiacarb, or you don't use any kind of snail or slug baits? Okay. And then once you're finished with that, please answer how satisfied are you with the performance of those baits that you do use, or other control method that you're using, whether it's methiacarb or a bait.
All right, and it looks like we have the majority of people who've answered, so I think Peter's going to close this one out.
Okay, so it looks like pretty evenly split between metaldehyde and iron phosphate. Few people use the sodium ferric EDTA, and less people use methiacarb. And surprisingly to me, about 30% of the people don't use any kind of snail or slug bait. Yeah, so using some other method, I imagine, for controlling their pests. And how satisfied? Looks like most people, let me go down, I can't, let's see if, oh yeah, I can. You know, most people feel that they're satisfied with what they have at this point. There's some people that are a little bit on the fence about whether they're satisfied or unsatisfied.
Okay, so I think he closed out of that, and then I think we can move on to Rory.
Okay, guys, thank you all very much for attending. A lot of the information that Cheryl has presented also applies to slugs, particularly the management side of things. So that's not something I'm going to delve into again here. Instead, I'm going to use my time to tell you a little bit more about slugs and their biology. We're going to take a look at some of the main pest species that you're likely encountering and how you can identify them. And I also want to talk to you about the ecology of one of the most damaging slug species and why understanding ecology is so important from a pest management perspective. So I'm guessing if I was to ask you all, what is a slug? Most of you would tell me it's a snail minus its external shell, and you'd be absolutely right. But from an evolutionary perspective, this might seem like a bad move. Surely losing a shell is not good for an organism that's comprised almost exclusively of water. But when you think about it, there's a number of big advantages to losing a shell. And they include the fact slugs can crawl through really remarkably tight spaces. Because they don't have the same calcium requirements as snails, they can also live in a much wider range of environments, particularly in areas that have acidic soil. And because they're not carrying a heavy shell around on their back, they can also move a lot quicker than snails. And the top speed for a slug has been clocked, would you believe, at 14.4. That's not miles per hour, that's actually meters per hour. So these things do not move very quickly. I know you all know what a slug looks like, but I do want to highlight some of the main body parts because I am going to be talking to you about them. I'm going to be mentioning them in the slides of this webinar. And also a lot of these body parts are very important when it comes to identifying different species of slugs. So we know the vast majority of slugs, they do not have an external shell. But surprisingly, many species have an internal shell, and this is what it looks like. It looks like a small oyster shell, usually located in this mantle, or this structure. It's called a mantle, and it looks like a saddle really at the front end of the slug. The mantle also has a breathing pore that's sometimes called a pneumostome, and that's always located on the right-hand side of the slug. If you're taking a picture of a slug to get an expert to identify it, it's very, very important that you take a photo of the right-hand side of the specimen, because the position of the breathing pore on the mantle is a very important feature when it comes to identifying different species of slugs. At the front end of the slug, and Cheryl had mentioned this when it comes to snails, we've got ocular tentacles, and as Cheryl said, these are used by slugs and snails to detect light. But recently, evidence has come forward that slugs and snails also use these structures to detect odors in the air, so they act a little bit like a nose as well. The lower tentacles, we call these the sensory or the peduncular tentacles, and the slug uses these to taste, so they act a little bit like a tongue. Other important features I want to show you today are the keel, and this is a raised ridge that runs from the back of the mantle to the tip of the tail. At the tip of the tail, we've sometimes got what's called a caudal mucus pore, and that's a large pore often filled with a thick mucus plug. We think the role of this mucus plug is in pre-copulatory behavior. When slugs are getting ready to mate, they chase each other around in a circle. And when the slug that's doing the chasing catches the specimen in front, we see it feeding on something in this region of the slug, and we're pretty certain it's feeding on the caudal mucus plug. The tubercles, to me, these looks like the building bricks of the body. And when slugs are getting ready to mate, their genitalia literally fall out of the sides of their heads through this genital pore. As Cheryl mentioned, snails and also slugs, they're hermaphrodites. They have both male and female reproductive organs. When it comes to identifying closely related species of slugs, the structure of these genitalia is very, very important. I'm not going to get into that with you today. It's quite technical and we don't have the time. But if any of you are interested in learning more about this, I'd be very happy to send on some literature. Please feel free to contact me. One of the things I've been doing since I moved to the US from Ireland about 10 years ago is I've been trying to get a handle on what species of slugs are causing the most damage to crops throughout the west coast of North America. Not surprisingly, it appears to be the invasives, and the vast majority are accidental introductions from Europe. Most of these slugs and also snails accidentally arrived here, and most of them on imported shipments of plants, others which I believe on imported shipments of tiles from the Mediterranean region, and others still on equipment that's being brought back to the United States from military bases in places like France, Spain, Germany, and Portugal. I don't have time to get into all of these species with you today. So what I've decided to do instead is to talk to you about some of the more important, some of the more interesting ones that you're likely encountering, and explain to you how you can identify them. So first up, we've got something called a banded slug. This is a species also known as the Valencia slug, because it's thought to be native to the Iberian Peninsula in Europe. It's very, very common in nurseries and garden centers throughout the West Coast, particularly in California, and it likes to feed on a whole range of ornamental plants. An easy species to identify, if you have a look at it, you can see there are two distinct tram-line stripes running the length of the body. That's a pretty good diagnostic trait for this species. And also the mucus is very, very watery. If you put a specimen of this slug in your hand and gently rock your hand back and forth, the slug will start to glide on its own mucus. And that's unusual for most species of slugs. If you were to do that with other species, the slug would just stick firm in your palm because its mucus is so sticky. Also, you probably noticed this hole in the mantle, and this is not typical for the species. It's mechanical damage, but inside you can clearly see it's an off-white color, and this is the slug shell that I spoke to you about earlier. Also with this species, we tend to find it in clusters. Where you find one banded slug, you tend to find a whole bunch of them clustered together. Next up, we've got something called a European red slug, and this is one of the largest invasive slugs we've got on the west coast. Again, a relatively easy species to identify. If you look at the front end, you can see that the head and tentacles are a contrasting darker color to the rest of the body. And also, it's got a very nice orange foot fringe, and this foot fringe has got distinct thin black lines. It's another good diagnostic character for this slug. We found this slug in a whole manner of disturbed areas in California and Oregon, and because of its size, it really has a voracious appetite for living plants. Here in Oregon, it's unusually common in campgrounds, and we think the reason for that is that campers are inadvertently transporting it from one campsite to another on the underside of their tents. Also, when it's disturbed, this slug sometimes rocks its body back and forth in a to and fro motion, and it almost looks like it's dancing. The cellar slug, this is one of the most common species that we encounter around urban homes and in urban gardens, where it tends to hang out in dark, damp places such as sprinkler valve boxes. It's a large yellowish brown slug, sometimes yellowish green, and it's got very sticky yellow mucus. But if you look at the front end of the slug, and the tentacles are a kind of a petrol blue color, and that's a good diagnostic character for this slug. In terms of food, this species loves to feed on pet food, particularly dog and cat food, and you may see it feeding in your pet's food bowl in the evening time. That's a behavior that we take advantage of in my lab when we're looking for specimens of this slug. We place some dog food around campus and around urban areas, and the slugs come out and feed it, and then we can bring it back to our colony or to our lab to start up some colonies. And the shell slug, this one is a real oddity in the slug fauna on the west coast of North America, because it's one of the very few slug species that actually has an external shell. It very rarely comes to the surface, this slug. It spends the vast majority of its time underground, and because it's a carnivorous species that feeds almost exclusively on earthworms. It doesn't feed on plant material, and for that reason, it's generally not regarded as an agricultural or a horticultural pest. But in saying that, we do not know the impact that this slug has on potentially beneficial earthworm populations. And I suspect that this species is probably quite widespread throughout the West Coast and probably throughout the nation. But because it spends most of its time underground, it's more difficult to find this slug, and it's probably under-recorded. So I've purposely kept the worst until last. The great field slug, not only is this the most damaging slug species on the West Coast, it's also one of the most damaging slugs found throughout the world. It lives in a huge range of environments, and it's a very serious pest of fruits, vegetables, seed crops, small grain crops, and ornamentals as well. And if you grow plants of any kind, I guarantee you've had this slug at some stage. Again, quite an easy species to identify, because when it's disturbed, it secretes a milky colored mucus. And you can see the milky colored mucus quite clearly on this darker colored specimen as the milky colored mucus there between the body tubercles. So because this species is one of the most damaging slugs we've got on the West Coast, particularly here in Oregon and in Washington, one of the things that my team and I have been trying to do over the past couple of years is to try and understand the life cycle or the life history of this species. And because in order for us to effectively manage any pest, we've got to know as much as we can about its biology. That's absolutely crucial. So we set up 16 slug blanket traps in a 4x4 grid in 6 annual ryegrass fields here in Oregon. We chose annual ryegrass because it's a very, very valuable crop in the Willamette Valley. And also it's a crop that the grey field slug causes huge amounts of damage to every year. And this is one of our fields. You can see a pink flag here, here, and here. They mark the position of our slug blanket traps, and you can see one of those in the bottom right corner. They're essentially a 1/2 meter by 1/2 meter square with what looks like industrial strength aluminium foil on the upper surface that reflects the sun's light and keeps the trap dark and damp. Underneath, there's black perforated plastic, and then inside the trap, there's a highly absorbent material. And slugs really like hanging out underneath these traps because they create a dark, damp environment, and that's the type of conditions that slugs really, really like. Okay, so what did we find? Well, we brought all of the slugs that we collected back to the lab where we weighed each and every single one of them, because we wanted to determine the percentage of newly hatched slugs, juveniles, adults, and mature adults at each of our collection dates. And we've weighed, would you believe, over 7,500 slugs to date, and we have found some nice patterns. If you take a look at the left-hand side of this graph, you can see in October, juvenile slugs, which are in orange, were by far and away the most common life history stage. But as the fall and the winter and the spring progressed, mature adult slugs, which are in yellow in this graph, became more and more abundant. And it is these mature slugs that really are the egg-laying machines. So from a control perspective, it's very important that we eradicate or kill these slugs before they oviposit their eggs. If we don't kill them, then the growers and farmers are going to have a significant slug problem come the fall and into the following spring. In terms of eggs, most were laid from February through April, with peak egg laying occurring in early March. And this was consistent over 2 years. So having this sort of biological information is very, very important, because different control strategies will have different levels of efficacy against different slug life history stages. So for example, we know that invertebrate natural enemies, things like ground beetles and the beetle that Cheryl showed to you earlier, prefer to feed on smaller slugs. So taking advantage of these natural predators which live in our environment during May and June and during September and October, when the smaller slugs are most dominant in the population, will likely give us the best results. Also, and because the grey field slug lays most of its eggs during the spring, if we can kill the adult slugs before they release their eggs into the environment, or the baby slugs shortly after they hatch, then we're going to have a smaller fall slug population to deal with. Why? Well, because the slugs that hatch from the spring-laid eggs make up the bulk of the fall slug population. And it's in the fall that these slugs cause the most damage. So that's my part of the presentation. I think there are also some questions that I have for you guys. Peter, perhaps you could bring those up.
Yeah, Peter's bringing that first one up now.
Great. So the first question is, what other gastropod management tools do you use other than the baits that Cheryl spoke to you about? So your options are biological control, which would be, for example, would be decolate snails. Barriers, a good example would be copper. If you use traps, such as bear traps, and hand picking. And if you use others, you can select that option. Or if you don't use any other management tools, there's an option there for you as well.
And we're going to bring up the results for this one.
Great. So some of you already do incorporate biological control, 23% of you. Almost a third of you also use barriers. Some of you use traps, but the vast majority of you use hand picking as a tool as well. Okay, very useful information. Thank you. So next question is, which do you consider more of a pest? Do you consider slugs more of a pest than snails, or snails more of a pest than slugs? Or do you think that both are more or less the same?
Looks like the majority of people have answered that.
Okay, most of you consider snails. Okay, great, very useful to know. So almost half of you consider snails to be more of a pest, 35 of you consider them to be more or less the same, and 17% of you consider slugs to be more of a pest. Okay, great, thank you.
And this last poll, I think, has 3 questions. He's going to bring that up. And so you might have to scroll down to see all 3.
Okay, these questions are to do with novel tools, and if novel tools were available, such as biological control, would you be willing to use them, and so on.
Okay, he's going to close this one out and bring up the results.
Okay, so if novel tools such as biocontrol were available, how likely are you to use them if they were same price as current methods? I think this is a bit of a no-brainer. Almost 3/4 of you said that you would try them. Same question, but if they gave more reliable control and were more expensive, now you're getting a little bit more hesitant. 58% of you would try, but over 1/4 of you would wait for others to try them first. And the last one, how much extra would you be willing to pay for new slug control tools? Almost half of you would be willing to pay 1.5 times as much. Approximately 20% of you would be willing to pay twice as much, and 14% of you would not pay more. Okay, this is very useful information for us. Thank you, guys. So we pass you over to Irma and to Adler now, who are going to talk to you about some novel tools that we're currently working on.
I will continue the topic on biological control, and I will talk about the findings that we have on phosphoroditis, and then try to be very brief about this because we have less than 15 minutes left. Biocontrol, as the name implies, is the use of living natural enemies in order to manage pest populations and reduce their damage. And this is usually accomplished using pathogens, parasites, parasitoids, and predators. Biocontrol has a long history in the US, and the first successful classical biocontrol took place right here in California, and that was when the Vidalia beetle was imported from Australia in order to manage insect pests in California citrus groves. So although there are advantages and disadvantages known for biocontrol use, there is a consensus that indeed biocontrol is useful and it's safer compared to chemical control. Some of the present-day examples of biocontrol that can be easily bought from nursery garden centers would be the ladybugs and praying mantis. And of course, we mentioned a while ago about decolate snails that are used in certain counties in California. And of course, the entomopathogenic nematodes. And having mentioned nematodes, then maybe we should refresh our mind on what nematodes are. They are the most abundant and the most genetically diverse multicellular organisms on Earth. And most of them are microscopic organisms that, like insects, undergo life stages from eggs to 4 juvenile stages until they become adults. So I'll be introducing phosphoroditis, which is a gastropod parasitic nematode. And during the process of, in the life cycle of this nematode, there are events that take place that will force them into a period of dormancy. And that is, for example, the absence of food or overcrowding. So like third or fourth stage, they can revert to a dormant stage, that is the infective stage of phosphoroditis. And in the presence of a cue of food, like a healthy slug, then they enter through the natural opening on the behind the mantle. And within 2 days, then there will be symptoms that are expressed. And probably 5 days, then the whole carcass will be consumed by the nematode, and the cycle is completed and repeated. So most of the time you will see in this picture here, thousands of adult nematodes that are feeding on the carcass after the slug dies. So this is now, we would like to say that indeed this is a very, is a better alternative than the chemical control. Although it is not a quick kill, it is safe generally for humans, for our pets, for the animals, and the environment in general. And therefore, they can be used in the organic production system. Likewise, in Europe, there is a record, or there are records of over 70% efficiency in horticultural and agricultural crops. And I will be showing some examples of that later on. In addition, they are also specific to gastropods, and therefore they do not impact other non-target organisms. And so they're not a threat to ecosystem diversity, unlike other predators or biocontrol agents that had been introduced to other islands that caused actually near extinction of other gastropods as well. Ultimately, this results in a stable and healthy crop environment, and thereby a safer product for all. One of the most questions asked by people who have heard about phosphoroditis is whether this is available in the US. It is not available in the US, but it has been marketed in Europe for over 20 years and used in 14 different countries. However, this nematode has only been found in the US recently, and therefore, if the company would have introduced this product into the US, then they would have introduced what would be an exotic species. So then, even until now, we do not have the commercial product. So in Europe, they have found efficiencies of this, whether in the field, in the greenhouse, especially for high valued crops like vegetables and ornamentals. And these are the different crops listed here that are examples of sensitive crops or suitable crops that had been protected by applying nemaslug. Hosta, for example, is a very susceptible plant, as you know. In addition to that, field crops and fruit crops are also protected from damage due to gastropods. And this is usually a recommended rate of 300,000 per infected juveniles per square meter. And in most of these research that have been published, the efficiency of nemaslug is comparable to metaldehyde or any chemicals that they have used as a positive control, and sometimes even better than metaldehyde. Outside Europe, the P. hermaphrodita is also commercialized as a slug tech by a company called Dudutech. And again, the target is for that to be used in the field or in the greenhouse. So maybe you'll be asking whether it will be really useful in the US, and why do we think it is going to be useful in the US? There are 4 reasons that I would like to present. And first is that we discovered not only one species, the P. hermaphrodita, that was commercialized in the UK, but there are multiple species that were found, and that's one is Californica, that was first discovered here in California, and then P. papillosa. And not only that they are distributed in California, they have been found in Oregon as well. And more importantly, they have recovered this Phasmarabditis hermaphrodita in the field. And so this means that the nematode is more probably widely distributed than originally perceived. Thirdly, there is a growing demand for safe and effective tools, and we have seen that in our prior surveys just a while ago, and especially for high-value crops and in organic farming. And fourth is that we have demonstrated through lab assays that this nematode has gastropod killing potential. So in the next few slides, I'm going to show that indeed the Phasmarabditis is able to kill 4 invasive slugs. We've tested them also on 4 different snail species. And the efficacy or efficiency is dependent on the species of Phasmarabditis as well as the gastropod species. And in addition, they are also safe to non-target organisms like earthworms and banana slugs. So now we have very little time left. Just to show you, for example, this shows us that there are two, for example, gray field slug and marsh slug are the more susceptible species or highly susceptible species. First mortality was observed either 2 or 3 days after exposure, with average mortality of 77% and 80% respectively. These are averages of 2 trials. And also Valencia slugs and black field slugs are the less susceptible species because then mortality was observed later and with a lower mortality of 40 and 45% respectively. As to snails, we have tested them on brown garden snail and giant African land snail and mortality was observed 5 days after exposure, but with a higher mortality of 80%. Another snail that was tested is amber or water snails that are not really considered as pests, but they are considered as nuisance pests or nuisance gastropods. And at the end of the 3-week observation, all of them died. When 3 species were compared against decollate snail, P. papayosa had shown even 1 week after exposure, P. papayosa was caused significantly higher mortality compared to P. hermaphrodita and californica. And at the end of the experiment, it was able to kill 70% of the population as opposed to P. hermaphrodita and P. californica with less than 10%. And like I mentioned, the Phasmarabditis hermaphrodita, the same isolate, did not cause any mortality on the 4 species of earthworms that we have tested, and of course the banana slug as well, as opposed to the positive control, which was gray field slug that were all, most of them died. Just to show you some symptoms of this infection on susceptible slugs, gray field slug and marsh slug, you can see the swollen mantle that Rory was talking about, the mantle. And then at the end, in 5 days, for example, you will only see the vestigial shell that is the remnant of the slug. And on less susceptible slugs, Valencia and black field slugs, they were quite surprised because black field slug is quite a hardy slug, but then still they were killed by Phasmarabditis. Just another slide to show you that indeed to demonstrate, for example, that 3 days after exposure, application of nematode at 1x or 5x protected the leaf from the damage, and especially for 5x, if you compare it to 0 nematode, then most of the leaves there are consumed by the slug. So the nematode is able to protect the leaves from damage due to the slugs because they've lost appetite after exposure to the nematode. Some of the snails we tried, and of course the non-target species, particularly earthworms and charismatic banana slug. So in conclusion, gastropod biocontrol using phosphorodiatis is in its infancy in the US. However, our current funding allows us to optimize phosphorodiatis bacteria partnership that is most virulent and would have a wider host range and of course safe to non-target organisms. And of course, the idea is to use the species that are already widespread in Western US. And we would like to think about Californica in that context. And then of course, the species that are best suited therefore to local conditions. Thank you for your attention. And now there will be some questions that we would like you to ask you about biocontrol and phosphorodiatis.
Yes, thank you. So yeah, Peter's bringing up that poll, and I think there are 3 questions in this one poll. Okay, he's going to bring up the results screen now.
Oh, thank you. Okay, so to summarize that, the question on whether you are convinced of the benefits of using phosphorodiatis to manage gastropods, most people, most participants say 64% say yes, and 32% say they need more information. And then as to whether they would be interested to use nematode phosphorodiatis for management of gastropods, a 61% majority wants to try, and 25% say that they would wait for others to try first. And there is nobody who said that they will not like to try it. And then at the bottom, I think, so if phosphorodiatis consistently controlled snails and slugs, how many or how much extra would you be willing to pay? 1.5 times as much. Majority of you said yes, 1.5 times as much. 18% say NA. 3 times as much, 2%, 5 times as much, 2%. So thank you.
So I will go to the question. So this first one is from Martin, and he's asked, and I think this was for Cheryl, has tolerance to baits been documented in mollusks?
As of right now, there hasn't been any documentation. There have been some questions people have asked me about that. And the bait that people were asking about is the metaldehyde. I've tried to see if the snails, this is for brown snails, were susceptible or resistant, I guess, to it, and I could not document that at all. I think that would have to be something, I don't know whether anybody's interested in doing it on a more granular basis, maybe on a molecular basis, but from my sort of quick tests, I haven't seen that.
Okay, and then Judy has a couple of questions. The first one is she's heard that Sluggo and other baits will kill earthworms. Is that true?
I don't know. I do not think it will. So I don't know about Sluggo and the other one. My feeling is that it will not impact earthworms. The other question that people have asked about though is metaldehyde. Will that impact earthworms? And there's been some research from actually the company that makes metaldehyde, where they actually essentially stuffed the earthworm with metaldehyde, and they did not find that it had an effect. So I don't know whether Rory or anyone else has any information about that.
Sure, I have some additional information
Okay.
on that. Here in Oregon, earthworms in many of our agricultural fields pull metaldehyde pellets into their burrows to feed on, because they're after the cereal attractant that's in the metaldehyde. The metaldehyde itself appears to have no impact on them whatsoever.
Okay, great. Thanks for verifying that.
Okay, and then another question from Judy. How long will slugs last without a food source? And if she removes all the crops in her raised bed but keeps watering it, will they survive, and for how long?
That's a good question, Judy. Slugs can survive, I would say, for at least a week without any food whatsoever. But one of the things we need to remember is that slugs will also feed on plant and fungal material that's in the soil. They do like to feed on green plants, but when they're gone from their environment, they will start feeding on soil to take out the microorganisms that are there, fungi, lichens, and so on. So really the only way to get rid of them quite quickly would be to use some sort of a bait. If you're reluctant to use something as toxic as metaldehyde, as Cheryl mentioned, you could use one of the iron-based bait. Iron phosphate, for example, is approved for use in organic systems, and it's harmless to humans, harmless to pets, and there have been no non-target effects to other organisms that I know of.
Okay, and Richard has asked if there's anyone in the UC system that can identify an unusual snail in Tulare County.
I would say, okay, go on, Rory.
I would say probably not, Cheryl. I don't know of anybody else. When I was at the UC system, I might have been the obvious choice, but I'm now at Oregon State University. So if Richard is still online, Richard, if you want to send me some photographs, I should be able to make an identification from a photo. And if not, I don't know, maybe someone at the LA County Commissioner's office, Cheryl. I don't know if you know of anybody else.
Right. Yeah, I would suggest the ag commissioners. Yeah, I'd send it to Rory just for fun.
Yeah.
And then, but I'd also send it to the ag commissioner's office, because if it is unusual, then they would have an official record of it, and it can move on to other areas for management.
Okay, and there's a few questions in chat. One from Stephanie: how effective are carabid ground beetles at controlling garden snails?
Well, I'm not so sure about garden snails. They have been shown to be pretty effective predators of slugs in Europe. And the thing we need to remember about ground beetles is that they're generalist predators, so they will also feed on other organisms. There are some species that are known to have a preference for slugs, particularly the grey field slug and the marsh slug. In my lab at the minute, I've got a postdoc who's looking at this interaction between ground beetles and slugs, and is dissecting out ground beetle guts. And she's actually going to screen the gut contents for slug DNA, so we can get very firm interactions between certain ground beetle species and certain slugs. And when it comes to snails, there are not very many ground beetles that I know of that feed on them. There are some specialist sylphid beetles that will feed on garden snails, but they're not very common. And again, they do feed on other organisms. So they have potential, but I guess the point I'm trying to make is that we don't have a lot of information on ground beetle predation on slugs in North America.
Okay, there's another question. How effective is acetic acid on snails?
If you're talking like the 20% acetic acid that's used as a herbicide, I think it would be pretty darn devastating to a snail. But if you're just talking about straight up vinegar, probably if it were directly applied, you might get something. I mean, keep in mind that these are like contact materials. So if the snail is sort of shell up, it probably would not have that much effect. You know, this is all guessing on my part, because, you know, we have the calcium-based shell. Who knows, right?
Yeah, exactly.
So yeah, I mean, I don't know.
Okay, so is phosma, I know I'm going to say this wrong, phosphorhabditis available, and if so, where?
So phosphorhabditis is, as a commercial product, is not yet available in the US. And so from what we really know from BASF is the European, UK-based company that commercialized this. They are still, I think, not doing anything about it, except that as far as we know, they are talking with USDA, probably about registering the product here. And Rory had reaction with one of the global manager there lately, and yeah, they don't say anything yet, unless Rory knows more.
No, that's all I know, Irma.
Yeah. Yeah, so we don't really know what their plans are in terms of marketing phosphorhabditis, hermaphrodita, or other species here in the US. But as far as we know, there are mainly two criteria for them to register this product, biocontrol product, here in the US. That means they would prove that this nematode is widespread in the area. And number two is that this nematode is safe for non-target organisms. So pretty much we are doing research for them if they want to market it in the US.
And does the nematodes, do they attack any insects?
So far it has not been shown to attack other insects. So yeah, like I said a while ago, they are more or less really gastropod specific. This particular species that we have recovered, we know of one other Phasmarabdites that was isolated from earthworm, and for which it doesn't have a name yet, but we have started work on that, and we know that they infect earthworm, but it is also an earthworm isolate. That's the other Phasmarabdites, but also it kills gastropods. It kills slugs.
And Judy is asking if it would be okay for Europe to ship it to us.
Because they have not permitted it yet, they don't have a permit yet, I don't think they will be able to ship that here. No, because yeah, I can only, for example, request that if I have now in the research, I am in touch with BASF. If we are to use that to compare that with our isolate, they will do it. But then it has to be, properly done, using all the quarantine procedures and, permits. Yes, it can be done, but it will be in quarantine. Yeah.
Right. Yeah. I mean, just to be clear, is that you can't order, the regular person on the street, the grower, whatever, cannot have it shipped to the U.S. because just like any biocontrol, it has to go through a number of steps before it can be released in the wild per se. So please don't try that.
Okay, and then there's just one more question from Linda. Has there been any studies about bird populations helping with control?
I don't know of any studies about it, but I think more general observations for sure. Birds are important natural enemies of snails and slugs, but I don't know of any scientific study which has actually measured their efficacy. I don't know if you know of any, Cheryl.
I don't, but I do have to say that rats love ground snails. So if you're willing to put up with rats, you'll get some pretty good control of your ground snails.
I do have one more question that just came in. So which snails are the edible ones?
Oh.
So my wife is from Spain, so she absolutely loves snails. So the brown garden snail, which is the one that Cheryl spoke about, it's a major pest in California. That's one of the species that's eaten in Europe. The reason it is a pest in California was because it was introduced to the state as a potential source of food, but the market really turned out not to be there. And one of the milk snails, Otala lactea, is also supposedly quite tasty as well. I just cannot bring myself to taste them.
Yeah, and I guess there's one. Is it Helix pomata?
That's right, Cheryl. Yeah,
right. And that's from what I understand, it's a little bit meatier. So if you want to go for it, feel free. There's plenty of ways to do it online to find how to cook your brown snails.
Yeah, just make sure that it's pathogen and parasite free. Never eat anything without properly cooking it.
yes.
Okay, I think that was our last question. So I want to thank everybody for attending, and then thank you to all of you guys for presenting. This was our first webinar with a panel, so thank you. I think it went well.